I am currently pursuing my master's degree from Banaras Hindu University. Today I will be teaching you glycolysis. So let's start. Basically, glycolysis is the primary step in the cellular respiration. The respiration process is divided into 4 steps, namely glycolysis. The second would be the link reaction, third is citric acid cycle, and the fourth one is oxidative phosphorylation. In this lecture, we will be covering the glycolysis part. So basically, glycolysis was discovered by 3 scientists, namely Engel, Payoff, and Pyruvate. And based on their names, this pathway is also called the EMP pathway. Engel, Payoff, and Pyruvate. The location for glycolysis is cytoplasm. And it is applicable for both prokaryotes and eukaryotes. Both prokaryotes and eukaryotes, the glycolysis takes place in cytoplasm. Glycolysis is a 10-step reaction in which the first 5 steps, energy is consumed, and in the other half of the cycle, energy is generated. So we will be studying the reaction step by step. Basically, partial oxidation of glucose is taking place in glycolysis. The first step, we will start from glucose. Glucose is our substrate. In the first reaction, the phosphorylation of the glucose is taking place. So, in this reaction, ATP is utilized. Phosphorylation of the glucose at the 6th position of glucose, at the 6th carbon. So, it will form glucose 6-phosphate. It takes place with the help of an enzyme called hexokinase. In the second step, isomerization reaction is taking place, and this step is irreversible and is a rate limiting step. Why rate limiting? Because this reaction depends whether ATP is available or not. So this is also called the rate limiting step. The second step would be an isomerization reaction where the glucose 6-phosphate would be converted into the fructose 6-phosphate. For phosphate, I am using P. So this reaction is catalyzed by an enzyme called isomerase. Here, glucose 6-phosphate is converted into fructose 6-phosphate. These two are the isomers of each other. Glucose contains aldehyde group and fructose contains the ketone group. The third step would be the phosphorylation of fructose 6-phosphate. So, fructose, it will form fructose 1,6-bisphosphate. What is happening in this reaction? Fructose is converted into fructose 1,6-phosphate. There is a phosphorylation at the first carbon. So, fructose 1,6-bisphosphate. This is isomerized by an enzyme called fructokinase. This is also an irreversible reaction, and this is also rate limiting step. Here also ATP is used. So, in the first step, 1 molecule of ATP was used, and in the third step also 1 molecule of ATP is used. Now we will continue. This is the first step. This is the first step. This is the second step. This is the third step, and here the fourth step. With the help of an enzyme called aldose, two 3-carbon molecules are formed. Glucose is a 6-carbon molecule. Glucose 6-phosphate is also a 6-carbon molecule. Fructose 6-phosphate is also a 6-carbon molecule, and fructose 1,6-bisphosphate is also 6-carbon. glyceraldehyde 3-phosphate. The isomerization step, the enzyme involved is ribose phosphate isomerase. In the other half of the lecture, we will be studying the energy producing steps. So in the second half of the reaction, the substrate would be glyceraldehyde 3-phosphate. Now we will be taking two molecules because the dihydroxyacetone phosphate is also converted into glyceraldehyde 3-phosphate. Here, in the sixth step, glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglyceric acid. This is happening with the help of an enzyme called glyceraldehyde 3-phosphate dehydrogenase. Whenever there is use of this term dehydrogenase, that means one hydrogen molecule is removed. So, the transfer of hydrogen molecule from glyceraldehyde 3-phosphate to NAD plus to form one molecule of NADH. As we are using two molecules of glyceraldehyde 3-phosphate, so two molecules of NADH would be produced. So this is also a reversal. The next step, 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceric acid. Here, one ATP molecule is produced as one phosphate group is transferred from 1,3-bisphosphoglyceric acid to ATP to form ATP. This is catalyzed by phosphoglycerokinase. So this is the sixth step of the reaction. This is the seventh step. Now, in the eighth step, this 3-phosphoglyceric acid is converted to 2-phosphoglyceric acid. Basically, what is happening? The phosphate which was present at the third carbon is relocated to the second carbon. That's why 2-phosphoglyceric acid. The enzyme involved is phosphoglyceromutase. In the eighth step, this is the eighth step. In the ninth step, this 2-phosphoglyceric acid is converted to phosphoenol pyruvate. This is done by an enzyme called glycolysis. Here what is happening? There is a removal of H2. In this reaction ATP was produced. In this reaction water is present. In the last step of glycolysis, there is the formation of pyruvate acid. Here, one phosphate molecule is transferred to ADP to form ATP. This is done by an enzyme called pyruvate kinase. The transfer of the phosphate molecule from the high energy compound to ADP is called substrate level phosphorylation. So here in the 10th step and the 7th step, the ATP produced is produced by substrate level phosphorylation. So this is the overall steps of glycolysis. Now we will be studying the summary of glycolysis. So in the phase 1, in the first and third step, ATP was consumed. So 2 ATP was consumed. In the sixth step, 2 molecules of NADH was produced. In the seventh and 10th step, ATP was produced by substrate level phosphorylation. So 2 ATPs was produced in each step, the total would be 4 ATPs. But the net ATP gain would be, as in the first and third step, 2 ATP was consumed, so the net ATP would be 4 minus 2 equals to 2 ATP. Alright, now we will be seeing the final products of glycolysis. Two molecules of pyruvate acid is formed in the 10th step. Then 2 molecules of ATP, net gain of ATP, and 2 molecules of NADH. In the oxidative phosphorylation, 1 NADH can produce 2 ATP, so 3 ATPs. So 1 NADH can produce 3 ATP. So 2 molecules will produce 6 ATP. So total ATP gain would be 8 ATPs. But by substrate level phosphorylation, there would be gain of 2 ATPs. That's all for glycolysis.